guys. In the last part, we started to study about how this biotechnology applied in it is applied. Then in the last part, we have studied how the biotechnology is applied in medicine. We have studied about human insulin or humulin, how to make humulin they made by the help of the company, Eli Lilly company from America. Now we are going to study about gene therapy. So what is gene therapy? It is used for the treatment of genetic disorder. It is used for the correction of genetic disorder. You might have observed some small baby, newborn baby born with some genetic disorder. Parents are worried about the genetic disorder. So, by the help of this gene therapy, we can correct the genetic disorder. That only we are going to study. You can understand by the help of an example. In 1990, in the year 1990, one four-year-old girl, she was having the problem of ADA deficiency. She was having the problem ADA deficiency. ADA, what's the full form of ADA? Adenosine deaminase. Adenosine deaminase. So she was having the problem with ADA deficiency. It means one of the gene is responsible to produce this ADA. ADA means one of the enzyme. So that gene is lack. Because of that, the enzyme also it's absent. So this ADA, it is useful for the immune system function. So what happens if this ADA is not there? The girl immune system is affected. Now we can understand, we can see there are some therapy, there are some treatment. They have given some temporary treatment. You can understand here there are some temporary treatment. One treatment you can understand, bone marrow transplantation. So first treatment you can see bone marrow transplantation. Bone marrow transplantation. And the second one you can say enzyme replacement therapy. Enzyme replacement means frequently they have to inject the enzyme. Then the next you can understand the ADA, that enzyme means that enzyme is injected to the patient body. So this treatment you can understand means this is temporary, means frequently we have to inject, we have to insert. Once it is finished, again the problem will arise. So this you can say temporary treatment. Now we can study one more treatment that also you can consider temporary treatment. What they have done, they have taken the lymphocyte from the 4-year-old child. That lymphocyte they have taken out. Lymphocyte. So lymphocyte is one of the type of cell. It is present in the blood. It's a type of WBC. So there are two different types of lymphocyte, B lymphocyte and T lymphocyte. So they have taken out this lymphocyte, and in the culture means in vitro outside they have inserted this ADA, the enzyme they have inserted. Then they have to insert this lymphocyte into the body. But what happens here, as we know, the lifespan of WBC is very less, around 13 to 20 days, around 2 weeks. After that it will die. So it's not immortal. Lymphocytes are not immortal. So frequently they have to do this culture and they have to insert. So we cannot say this also one of the permanent solving. This also one not a permanent problem, permanent treatment. So next they understood we have to make one permanent treatment. So they have done the permanent treatment. What they have done, what we have to do means at the time of embryological stage we have to take we have to insert the particular gene into the baby. Particular gene at the embryological stage we have to insert the gene into the baby. Then permanently we can solve the problem. That only we can understand about the gene therapy. This is one of the important topic for the board as well as for the NEET exam. So here we have understood there are three treatments: temporary treatment and only one permanent treatment that you can understand, inserting the gene into the embryo. That only you can understand about the permanent treatment. So this is about the gene therapy. Next we can study about molecular diagnosis. Molecular diagnosis. diagnosis. So this also one of the application of biotechnology in the medical field. What happens, as we know, early detection of disease, early understanding of the particular pathogen, and early diagnosis is very useful, very helpful for the treatment of disease. As we know, some of the infectious disease, that is because of the pathogen. Once the pathogen is entered into the body, after some time, means it will be multiplied. After that, it will cause symptom of the disease. Then only we will go to the hospital and we will search about the treatment. That time what will happen? The pathogen, the concentration of pathogen will be more in our body. Then the treatment also will be late. It will cause some other severe problem. So early diagnosis is very important for the treatment of disease. So now here we can see there are three diagnoses, early detection of the disease by the help of biotechnology. That is, here you can see by the help of biotechnology, the techniques are PCR and ELISA test. PCR and ELISA test. In the last chapter, we have studied about PCR. What is PCR? It is polymerase chain reaction. So by the help of polymerase chain reaction PCR, we can detect the microorganism, the pathogen it is present in the body. So how we can understand by the help of PCR what happens here? By the help of this PCR, they will amplify the nucleic acid of the pathogen. PCR is used to amplify the nucleic acid of the pathogen. Immediately we can identify if it is there, the pathogen is there in the very low concentration in the body, then also we can find by the help of this PCR. So it is very helpful for HIV patient and in the cancer patients. It is very helpful. So the another method we can study in the next part. Please listen the next part also. Thank you, students.